Hey everybody, this is Dr. New, aka James R. Neuenschwander, MD. I'm the president of the Medical Academy of Pediatrics and Special Needs, or MAPS, and this lecture is to give you a leg up on the upcoming conference for those of you that are new to MAPS. This will give you a little bit of information. So let me get into presenter mode here. Okay, so this is concerning our conference that's coming up in the middle of March. I'm going to start with a little bit of overview and foundational principles of integrative medicine. So, a lot of our prior participants to MAPS had expressed concern that that initial day was in the foundations matrix was just like drinking from a fire hose. It's particularly true of people for which integrative medicine or integrative practices and those principles that are involved with it are foreign to them. So the purpose of this presentation is really to provide an orientation for that foundations course and make it a little bit easier to drink from that fire hose. You'll still be drinking from a fire hose, but this will make it a little bit easier. So let's start with a few principles. I mean, principle number one, and I always consider this the most important principle. Our bodies are designed to be well. Our job as practitioners is to remove the barriers for them being well rather than try and push them into a state of wellness. Principle two, we should be always asking what needs to be removed, usually some type of toxicity, and what needs to be added. This would be nutrition, supplements, mental, spiritual support to help heal the system. Principle number three, symptoms are almost always the body's attempt to heal itself. So be careful when you're treating symptoms because you may actually be delaying or interfering with that healing process. Always, always, always look for that underlying cause, that root cause that is creating the symptoms. My joke is always person goes to the doctor and says they're tired, and the doctor does a blood count and says, "Oh, you're anemic. Let me give you some iron for that anemia. It'll probably help your fatigue." And then the patient says, "Well, what about this bullet wound I have in my liver over here? Could that be contributing?" Yeah, maybe that's the reason why they have low iron in the first place. All right, principle number four, anything you do to suppress a bodily function is going to result in the body upregulating that process. And the opposite's true if you're trying to stimulate a function, the body will downregulate that process. I mean, this is one of the problems with the pharmaceuticals that we use. The body will always try to overcome what you're doing with that pharmaceutical. Principle number five, all pharmaceuticals have toxicity. All of them, without exception, have toxicity. And almost always, the long-term use of that pharmaceutical results in that toxicity outweighing the benefit of that pharmaceutical. So very few pharmaceuticals we've ever used have helped us live longer, even things that should be life-saving. So it's a big difference between using antibiotics for 7 to 10 days and taking a statin for 30 years. Principle number seven, almost all chronic conditions can be viewed through the lens of a system with some type of impairment being presented with an insult, an environmental insult that is greater than what that system can handle. I know it's a mouthful, but really that's what we're talking about. Toxicity that exceeds the body's ability to tolerate that toxicity usually is going to result in some type of illness. And then the final principle, the key to treating chronic conditions is to understand the fundamentals of how our bodies work and the importance of systems biology and whatever it is we do. All right, so we're going to start with some of the basics of cellular metabolism. Sorry, I don't want to give anybody flashbacks to high school biology, but here you go. Here's a cell. There's a bunch of different structures within the cell. There's a whole universe inside the cell. We all know about the nucleus. We know about the mitochondria. Probably we know about the smooth and rough endoplasmic reticulum. That's where we translate the DNA and we make those proteins. The ribosomes are little ticker tape things that convert messenger RNA into proteins, and then those proteins are folded within the ribosomes. They go over to the Golgi apparatus that packages things up and sends it out. But we also have things like the peroxisomes. The peroxisomes are where we, or about peroxisomes and lysosomes. So lysosomes are where we break down things we don't want, but peroxisomes are where we actually break down our fatty acids. You know, if you want to take your fatty acids and get them ready for the Krebs cycle, you need peroxisomes to break them down into those acetyl-CoA chunks. Peroxisomes are also the place where we make those fatty acids, where we take a 24, 26 carbon fatty acid and we chop it down to, that 22 carbon DHA or the 20 carbon arachidonic acid or whatever it is we're using. So just understand the cell is a very complex environment. Just one cell is an entire universe. We'll start with the nucleus of the cell. I mean, we know about DNA, codes, for the genes, codes for the proteins that we need. But the reality is most DNA is non-coding. 80 to 90% of our DNA does not code for proteins. The function of that DNA remains a mystery, although there are some theories on that. The important thing to understand about the DNA is that the expression of our genes, a process that we call epigenetics, is as important or even more important than the genes themselves. It's not the gene. It's not that you have a gene that puts you at risk for something, is how is that gene expressed in the environment that you're in. And the important thing about these epigenetic changes is even those can be inherited in the same way that DNA can. And so those epigenetic influences can be passed on to the next generation. The mitochondria, I mean, we know the pyruvate from fermentation in the cytosol of the cell, the fatty acids that are broken down in those peroxisomes, and amino acids, particularly glutamine, from cytosolic fermentation. You can ferment glutamine in the cytosol without the mitochondria. But all of those things will enter the mitochondria and be converted into acetyl-CoA. So acetyl-CoA is fed into the Krebs cycle to make NADH and FADH. Those are very high energy compounds, and you typically make one ATP with a run of the cycle. And then the NADH and FADH are fed into that electron transport chain. And when you combine that with oxygen, that efficiently makes ATP. That's why we breathe oxygen. It's one of the big reasons why we have mitochondria. Now, I apologize for the simplicity of this. I just want to make sure everybody is up to speed on what we're talking about. So this is a visual of that. You have your glucose. Whoops. You have your glucose being converted into pyruvate in the cytosol. That gets fed in. You have your fatty acids coming in, your peroxisomes being converted into the acetyl-CoA through fatty acid oxidation, beta oxidation. These all get fed into the Krebs cycle here, and the Krebs is going to kick out NADH, which goes to complex 1. It's going to kick out FADH, which goes to complex 2. And by the way, complex 2 is actually part of the Krebs cycle. That succinate dehydrogenase is actually part of the Krebs cycle. But as those electrons get shuttled from complex 1 and complex 2 to complex 3, you start pumping hydrogen ions out into this intramembrane space. Eventually, all those hydrogen ions come back in, and you create ATP. The oxygen itself is used in complex 4. That's where you're converting oxygen into water. And that's also the place where you can develop free radicals. So the mitochondria are not just the energy producers for the cell. They also regulate the cell cycle. They determine when a cell is going to undergo autophagy, which is recycling, or apoptosis, which is programmed cell death. So the mitochondria have different states in which they function depending on where the cell itself is at from a state of homeostasis. When it gets knocked off of that and gets injured, the mitochondria go through transitions and have actual different forms. And the mitochondria help the cell recover from that injury. And persistent injury to the mitochondria results in many of the symptoms we see in chronic disease, particularly the brain dysfunction, the aching, the fatigue. All those things are related to the mitochondria being stuck in that process. Membrane chemistry, I mean, this is probably the most important thing most of us never learn about. Membranes regulate pretty much everything in the body. A lot of this chemistry in our body happens on a membrane. We just talked about the electron transport chain. That happens on the inner mitochondrial membrane. That's very important. But the membranes provide structure to the cells. That picture I showed you and all those organelles inside the cell are defined by a membrane, and they separate those organelles from other things in the membrane. So you have mitochondrial membrane, endoplasmic reticulum membrane, nuclear membrane, Golgi membrane, peroxisome membranes, and on and on. So membranes also control that internal environment in the cell, and they include an electric potential. So again, a cell membrane will separate charge from inside the cell to outside the cell, and inside the cell, the charge is slightly negative. So we define cell death as when the charge across that cell membrane is zero. When it's zero, the cell is no longer alive. So a lot of that chemistry happens on that membrane. We talked about energy production, but protein production happens there, protein folding, nerve conduction. It's critical for cellular detox. You want to send off packets of information. You need functional membranes. So that chemistry is very, very important. Membranes are primarily made of phospholipids. I mean, they're both phosphatidyl types of phospholipids and plasmalogen types of phospholipids. And this is where, when you talk about the essential fatty acids and the fatty acids like DHA and EPA, this is where we use them. We plug them into these phospholipids to give different versions of them and different activities. So we use that DHA, EPA, arachidonic acid, oleic acid, along with cholesterol, solid fats, proteins, glycoproteins, all of that make up a cell membrane, which again is a very complex structure. So this is a picture of it. You know, you've got some pores here. You have proteins that go all the way through the membrane. They allow, ion transfer channels, that sort of thing, that allow things into the cell. You have this lipid bilayer. I mean, this is what phospholipids do. You have this phosphate head, which is water soluble. You have the lipid tail, which is hydrophobic. So automatically the tails are going to join to one another, and the heads are going to be outside and inside the cell. That makes up the majority of the structure. But again, most of us know you can get a bottle of phosphatidylcholine. It's liquid. If we didn't have something to hold together the structure of the membrane, we'd just be a puddle of oil. So within that membrane, we have lipids like cholesterol. We have also big lipids like sphingomyelin that help provide structure. And then there's also proteins within the wall that are either external, internal, or span the membrane. And then some of these have other things attached to them on the outside that is involved with cell structure as well. So again, very complex involvement of the cell membrane, very complex chemistry going on there. It's not something we're going to cover in detail in the foundations course, but you just want to be aware of the structure of a membrane. All right, we're going to move on to some biochemical processes that are very important, which you may or may not know about. First is methylation and glutathione. So methylation this is a methyl group. It's a carbon and three hydrogens. You attach that onto something that's methylating that product. If you remove it, that's demethylation. So why does that matter? The function of the methylation cycle is to generate this compound, S-adenosylmethionine, or SAM. SAM turns out to be the primary, not the only methyl donor for the body, but the primary methyl donor for the body. So methylation is involved in the expression of DNA, the conversion of serotonin to melatonin, the breakdown of your monoamine neurotransmitters like dopamine, norepinephrine, serotonin, the metabolism of steroid hormones, the process of detoxification, and 80 plus other reactions. It's a very, very important foundational process that is involved with health. And a lot of our kids with chronic illness have problems with methylation. So this is the methylation cycle. It starts with homocysteine. Homocysteine gets converted to methionine. For that step, you need methyl B12. Methyl B12 gets its methyl group from methylfolate, which gets it ultimately from an essential amino acid. From methionine, that gets converted to SAM. Again, that's the big methyl donor. So SAM, when it passes its methyl group onto one of these methyl transferases, I just have it shortened as an MT there. It transfers the methyl group to a methyl transferase, and it's going to use it in whatever biochemical process you need it for. And in the process, once SAM loses its methyl group, it becomes SAW, S-adenosyl homocysteine, and then that gets recycled back to homocysteine, and it starts all over again. So The other big thing we worry about is glutathione. So glutathione is a hugely important compound, and this is something that most doctors have never heard of. I mean, the big thing we need glutathione for is to breathe oxygen. When we use oxygen in metabolism, we create oxygen-free radicals. If we did not have glutathione, well, there really would be no way to put those out. So it's also very, very important in phase two liver detox, and this includes the glutathionase transferase enzymes, the GST enzymes, that are heavily involved in detoxifying metals like lead, mercury, arsenic, cadmium. We need glutathione for that. Glutathione is also one of the only molecules, methylene blue might be another, that doesn't become an oxidant when it reduces something. So most, vitamin C, it reduces something, it becomes oxidized itself. Glutathione does not because of the unique structure of glutathione. So this is the basic glutathione reaction. You have glutathione, reduced glutathione, which we shorten as GSH. It runs into a free radical. It then reduces that free radical, so it's no longer dangerous. And in the process, instead of being GSH, it binds two oxidized glutathione molecules bind to each other and become GSSG. So it's not going to go beyond that. That's not going to go oxidize anything else. So that's what is unique about its structure and prevents it from becoming an oxidizer. The other thing is the GSSG, the oxidized glutathione, has to be recycled back to GSH, and that uses an enzyme called glutathione reductase and something called NADPH, which is another high energy compound, as the electron donor to do that. So here you have your oxygen-free radical, and this is what the body does with it. So the oxygen-free radical gets converted into peroxide using an enzyme called SOD. Then from peroxide, you have to break that down in one of three pathways. One is using catalase, which will convert it to water and oxygen. The other is using glutathione with this glutathione peroxidase enzyme, and that's the primary pathway that we're going to use. And then the third one, if you don't do that, it can be oxidized by either copper or iron in a +2 form and create this hydroxyl free radical. If it goes down that pathway, the hydroxyl free radical is actually more toxic than the oxygen free radical. And that's why glutathione and having adequate catalase is so important. So the things that create these oxygen free radicals, the biggest one is our mitochondria, just using oxygen to make energy. It's never 100% efficient, and you're going to have some leakage of oxygen that escapes as that O2 minus. I mean, that's an oxygen that only has 7 electrons in its outer orbit. It's going to want to steal an electron from somebody, and that's an oxygen-free radical. The second source is from hypoxia. We generate something called xanthine oxidase to try and deal with that, and in the process, you develop oxygen-free radicals. And the third is from this NADPH oxidase, which gets activated by the immune system when we have infections and that sort of thing. But again, the primary source is going to be from mitochondrial leakage of those oxygen-free radicals. So glutathione is actually associated with methylation. So here's our methylation pathway. When you look at that homocysteine, that's actually a key element. It can either be methylated into methionine or it can be transsulfurated into something called cystathionine. Which direction that goes in, whether you methylate or you sulfate, is dependent on the state of the cell. If the cell's under stress, it's going to go down the cystathionine pathway. That gets converted to cysteine and also ultimately gets converted into reduced glutathione, which the cell can then use to recover from whatever the insult might be. So a lot of children with chronic illnesses have genetic issues with one or both of these pathways. These are not mutations. We call them SNPs. They're variants, kind of like having blue eyes. We don't call a person with blue eyes a mutant, but it is definitely a variant from what most people have. So it's the same thing here. There are different levels of your ability to make glutathione and use glutathione, the efficiency of the enzymes that use glutathione, all that stuff's genetic. And a lot of kids that have chronic illness have glitches in those pathways. And it means that they don't detox very well. They don't handle oxidative stress very well, and they have problems regulating both their body chemistry, brain chemistry, and their hormones. So these issues give rise to symptoms. We label those symptoms as asthma, autism, ADHD, OCD, anxiety, et cetera, et cetera, et cetera. All right, we're going to move on to the gut and the gut-brain immune axis. If you're new to integrative medicine, this is really the heart of a lot of what we do, is working on these things. So I'll try and go over this briefly. We'll cover it more in foundations. And again, once you are into the fellowship program, we get into the nuts and bolts of this. So here's some gut basis. I mean, we have classic functions of the gut, which everybody knows about: the digestion and absorption of nutrients, the protection against pathogens in food and water, and the elimination of some of those endogenous toxins, the one we produce internally. But the non-classical functions, regulating the immune response, alteration of autonomic function. Again, our sympathetic parasympathetic nervous system is heavily influenced by the gut. It provides barrier against antigen exposure. It produces neurotransmitters. Most of the neurotransmitters we have in our body come from our gut, not from our brain, and it's also involved in detoxification. So the gut has this dual role. On the one hand, it has to absorb essential nutrients. On the other hand, it has to stop pathogens and large proteins that would induce an immune response from getting absorbed. So you have to have that semi-permeable membrane lining your gut. So the bowel mucosa that makes up that membrane is a combination of enterocytes. That's what most people would think of as, the normal intestinal cells with the brush border and the things that actually absorb the nutrients, but also goblet cells that make mucus, Paneth cells that make antimicrobial compounds. If you ever measure a calprotectin in a stool, that's actually a antimicrobial protein that's made by a Paneth cell. You have these neuroendocrine cells that make neurotransmitters, and they also make neuroendocrine hormones. And then you also have these intraepithelial lymphocytes, because there's a huge immune component to the gut as well. So the structure of the bowel means that the gut is intimately connected with the function of the immune system. So if you change the gut environment, you change the microbiome, you're going to change that immune function, good or bad. So again, when we're looking at autoimmunity, recurrent infections, allergies, we're always looking to the gut to see, is there a way to balance that better so that we can improve outcomes? And the structure I just discussed doesn't actually include the microbiome. This is the ecosystem of organisms that live in our gut and lives in our skin. There's all kinds of ecosystems in our body for these microorganisms. And these are very, very important. I mean, it's thought that the number of bacteria on and in our body outnumber our cells by at least a factor of 10 to 1. That's an estimate. It may be more, it may be less, but the bottom line is they're very, very important. And there's a continuous feedback between those bacteria and the lining of the gut, and particularly those immune cells that are lining the gut. But there's also feedback between them and the nervous system of the gut. So the gut is connected to the brain directly through that autonomic nervous system. So the gut has input from both the sympathetic and the parasympathetic, but the big one is really the vagus nerve. From the distal esophagus all the way down to the descending colon, that's all governed by the vagus. And that includes the pancreas and the gallbladder, digestive enzymes, stomach emptying, motility, all those things are affected. The really important thing about the vagus is that the importance of the vagus isn't so much from the brain down to the gut. That's maybe 20% of the effect of the vagus. It's the opposite. 80% of the function of the vagus is from the gut to the brain. So it's actually providing information to the brain, to the central nervous system, about the state of the gut and what needs to be done. And what's really cool is output through the vagus and through the sympathetics will actually alter the immune response, can actually alter that microbiome. So the gut and the microbiome make compounds that directly or indirectly impact brain function. So we have short chain fatty acids, things like butyrate or propionate. Butyrate will help your brain. Propionate might make you a little bit irritable. The gut makes neurotransmitters. Again, 80% of all the 90% of all the serotonin we make comes from the gut, not from the brain. And similar numbers are true of dopamine and norepinephrine, some of the other ones. And then the gut also makes endotoxins, right? So we have fermenting bacteria, anaerobic fermenting bacteria and yeast that make these endotoxins that can get absorbed. And if you don't think endotoxins have an impact on the brain, I just want you to think about alcohol. That is an endotoxin produced by a certain type of yeast that, we like to drink that stuff because it definitely alters how our brain functions. Finally, the microbiome of the gut is involved with the process of detoxification. Now, there's not as much written about this, but we know microbes in the soil can detoxify whatever you put on the soil. Farmers know this. You got to have a good, healthy bacterial ecosystem in the soil if you want to detox whatever's happening in the soil. Well, it's the same with our gut. And these bacteria have enzymes. They may outnumber us 10 to 1 or something like that, but their DNA diversity, the diversity of the DNA may be 100 to 1 or even 1,000 to 1. They have genes we don't have. They have enzymes that can break bonds we can't break. And the classic one is the carbon halogen bond. We don't really have an enzyme to do that, but the gut bacteria do. And the gut bacteria, by the way, can also detoxify pharmaceuticals. So the state of that microbiome can affect how well your prescription is going to work. All right. So again, the microbiome has a direct impact on the vagus nerve. It directly impacts the vagus nerve interaction. It affects neurotransmitter production. It directly produces short chain fatty acids. It creates these neuroactive toxins. Its indirect activity is going to be through detoxification, and that's going to affect how we feel and how our body functions, both our immune system and our brain. It's going to modulate the immune system. Again, there's a direct connection. The saying is 80% of your immune system lives in your gut. So you change the gut, you change that immune response. And then it's also going to alter the medications. And I would also say the nutraceuticals that you're using. It's going to alter those because of how it detoxifies things. So I'm going to finish off with this concept of systems biology because this is really a different way of thinking. When we learn biochemistry, we learn physiology, right? We learn the Krebs cycle, we learn the uric acid cycle, we learn, individual reactions, and we learn physiology. We learn, oh, this is the cardiovascular system, the renal system, this is the neurologic system. What they don't teach us is this idea that in biology, I always say there's no little arrows inside the cell that tell a compound where to go, right? In biology, it's systems. Everything is connected to everything else. And the reason why that's important, it's not just a concept, but when you're doing something, whatever you do, good or bad, whatever you do, one modification is not just going to have one impact. It's going to change the fabric of the whole system. And you have to keep that in mind when you're intervening with something. So unfortunately, this is the complete antithesis of our pharmaceutical model, right? So in the pharmaceutical model, you have a symptom, we have a pharmaceutical. So symptom, treatment, symptom, treatment. There's little consideration for how this symptom might be connected to other symptoms and how these treatments alter everything else, right? So because of that, is maybe a problem with these long-term use of the pharmaceuticals. Almost none of these things have improved life expectancy. And there have been a number of medications that we have used in the past that actually shortened life expectancy, like particularly blood pressure medication, where, lowered the blood pressure, great, but you didn't live as long because of the toxicity of the pharmaceutical. So systems biology would dictate looking at a patient's symptoms through the lens of everything else. How is this connected to that rather than just one organ or one biochemical pathway? So, example here, if a child has a runny nose, the problem usually is not with the nose. It's an issue with the health of the mucosa, the function of the immune system, the food and environmental exposures, and the health of the gut. You have to think of all those things. Kid comes in with a runny nose. Well, your next question is, how's he eating? What's his gut function like? Is he sleeping? Is this something that happens all the time? Is this just once in a blue moon? Where has he been? What's he been exposed to? On and on and on and on, because all those things get to the root cause of why that child has a runny nose. So systems biology would also suggest that if you're having problems finding the answer to a question, frequently it's because you're not asking the right question. And this is always a good thing to think of when you get stuck. This happens to me all the time because I have an allopathic brain and I was trained as an allopathic physician. So I'll run into a situation where I can't come up with an answer and I have to take a step back and say, whoa, whoa, whoa. Maybe you're asking the wrong question. And the classic example is you have a patient clearly has yeast in their gut. You're doing this yeast program, that medication, this, that, the other, and they're not getting better. The question isn't what's the next antifungal thing I can do? Do I need to do a stronger antifungal, a different antifungal? Do I need to do it longer? Maybe the question you should be asking is, Why is the yeast there in the first place? All right. That's much more of a systems biology approach to the problem. So always remember, and I just want to finish with this because I started with this, I'll finish with this. This is principle number one. We are designed to be well. If you have somebody sitting in front of you who's not well, something is stopping them from getting to that state of wellness. Your job is to figure out what's preventing that, what that body needs, what barriers there are, remove the barriers, feed in the stuff you need, and see if you can get that child into a state of wellness. So with that, I will complete this and we will see you in Myrtle Beach. I am really looking forward to it.